Dead Sea Scrolls.
Dead Sea Scrolls.
Dead Sea region.
One of the first results of this amazing research.
Portions of the Dead Sea Scrolls are here at DC's Museum of the Bible, removing all obscurity and open for all to see.
For 70 years, the Dead Sea Scrolls were hiding a secret nobody thought to look for. Everyone kept circling the same question: what do these ancient texts actually say? Scholar after scholar, generation after generation, treated that as the only question worth asking. Almost no one paused to ask the other one, what are these things physically made of?
This is a sample of one of the big scrolls. We have 5 of them, really well preserved.
Then, in a lab at Tel Aviv University, a molecular biologist named Oded Reshavi extracted ancient genetic material from dust taken off a single fragment, ran it through a sequencer, and froze. The animal whose hide had become that piece of parchment could not have lived anywhere near the cave it was found in, not for a season, not even for a single day. And that one impossible result was about to unravel everything scholars believed about the most famous manuscripts on Earth.
These are the earliest copies of the Bible that we have. They're older than anything that we had previously.
This sample comes from one of the major scrolls. There are actually 5 of them, astonishingly well preserved for objects that spent centuries in a cave. To understand why that one result mattered so much, we need to go back to where this whole story begins: a hillside above the Dead Sea, and a shepherd who had no idea he was about to rewrite history. A stone thrown into the dark. Back in 1947, a young Bedouin shepherd named Muhammad Ed-Dib tossed a rock into a cave near the shore of the Dead Sea while searching for a goat that had wandered off. He heard something shatter. Curious, he climbed inside and discovered ancient clay jars, sealed for centuries, holding leather scrolls wrapped carefully in linen. He had no clue what he'd stumbled onto. Nobody did, not yet. He'd simply been chasing a lost goat. When he reached the mouth of the cave, he threw a stone in just to spook the animal out. Instead, he'd walked straight into what would become the most important archaeological discovery of the 20th century, the oldest known biblical manuscripts in existence, some more than 2,000 years old. Between 1947 and 1956, roughly 25,000 fragments, pieces of nearly 1,000 separate manuscripts, were pulled from 11 caves near the ruins of Qumran. Among them were the earliest known copies of Genesis, Isaiah, and Psalms, along with sectarian rulebooks, hymns, and apocalyptic visions. The texts date from around 250 BCE to 68 CE, arguably the most chaotic period in the history of both Judaism and the earliest Christian movement. Hidden away in the dry Judean Desert, these scrolls had quietly survived the fall of the Second Temple, the collapse of Rome, every empire that rose and crumbled above them across 2 millennia. But when scholars finally cracked them open, the texts didn't hand over easy answers. They handed over questions that would take 70 years just to begin untangling. Where did they come from? Who wrote them? Why hide them in this specific corner of the desert? For most of those 70 years, scholars thought they'd figured it out. They hadn't. The theory that held for decades. Researchers from institutions across the globe studied every visible clue these fragments offered, armed with nothing but their eyes, their training, and their instincts. They compared handwriting letter by letter. They studied the color, texture, and thickness of the parchment, hoping to trace fragments back to a single animal. They cross-referenced language, theology, calendar systems, and literary style, hunting for any pattern that might connect the pieces. These remain the oldest surviving biblical manuscripts anywhere, older than anything scholars had ever laid hands on before. The dominant theory pointed to the Essenes, a Jewish sect that ancient writers like Josephus and Pliny the Elder described as a secluded, monastic community in the Judean wilderness devoted to ritual purity, scripture copying, and waiting out divine judgment on a corrupted world. The ruins at Qumran, right next to the caves, matched the kind of settlement Josephus had written about centuries earlier. The pieces seem to lock into place almost too perfectly. The sectarian writings described a closed religious community with its own leadership, its own laws, and a solar calendar that clashed with the lunar calendar used by the priests in Jerusalem, essentially a theological middle finger to the temple establishment. The apocalyptic texts were saturated with hostility toward that same establishment. The War Scroll, which lays out a final cosmic battle between the sons of light and the sons of darkness, reads exactly like something a breakaway sect would write to make sense of its own exile from mainstream Jewish society. The conclusion felt watertight. The Essenes lived at Qumran, copied their scriptures there, and when Rome crushed the Jewish revolt in 68 CE, they hid their library in the surrounding CE8 CE to protect it. For decades, that was the accepted story, taught in seminaries, showcased in museum exhibits, repeated in documentary after documentary. The Dead Sea Scrolls were simply the Essenes' personal collection. Case closed, nothing more to debate, except stick around, because all of that is about to fall apart.
He was looking for a lost goat when he came to the entrance to a cave. He decided to throw a stone into the cave to see if he could flush out the goat.
Cracks nobody could close. The Essene theory dominated for 70 years, but underneath it, uncomfortable details kept surfacing. Details the tools of the era just weren't equipped to explain. The scrolls showed wild, almost baffling inconsistency from text to text. Different versions of the same biblical book sat next to each other. Different handwriting, different scripts, different levels of skill. Parchment quality ranged from expertly prepared skins to rough, almost sloppy material, sometimes found sitting side by side in the very same cave. When radiocarbon dating finally got applied to a batch of fragments in the early 1990s, some scrolls turned out to predate the Qumran settlement itself. The timeline simply refused to line up the way the theory required. And nobody could crack the bigger mystery underneath it all. Why would one isolated, tightly disciplined sect keep multiple, conflicting versions of its own holy texts? Why hang on to two entirely different editions of the Book of Jeremiah, one noticeably longer than the other, with whole passages missing and chapters rearranged, inside a community supposedly built around strict religious uniformity? A handful of scholars floated alternative theories on the margins of the field. Maybe Qumran wasn't a monastery at all, but a pottery workshop, a winter estate, a military post. Maybe the cave served as a sacred storage site where worn-out texts from many different communities were brought to be respectfully retired rather than discarded. These ideas existed, but none of them carried the hard evidence needed to knock over a paradigm that had stood firm for generations. Then, the breakthrough arrived from a direction nobody in the field expected. Not an archaeologist, not a theologist, not a theologian, a geneticist over lunch. The lunch that changed everything. Tel Aviv University, molecular biologist Oded Reshavi sat down with biblical scholarship had never fully Reshavi sat down with biblical scholar Noam Mizrahi. Their conversation wandered, the way chats between longtime colleagues tend to, toward the frustrating puzzle of matching up thousands. Mizrahi spelled it out plainly. 25,000 fragments, no titles, no page numbers, no reliable way to connect one piece of parchment to another beyond comparing handwriting and guessing at how old the skin looked. Methods that were subjective, imprecise, and could go badly wrong without anyone even noticing. Reshavi listened carefully. Then he asked one simple question. What are the scrolls actually made of? Parchment, Mizrahi answered. Animal skin. Reshavi went quiet for a moment. Parchment has DNA. That single sentence cracked open a whole new field of study that nobody had thought to pursue before. Reshavi specialized in ancient DNA analysis, a technique that had already reshaped the study of human prehistory, tracing migration and population history across tens of thousands of years. Sitting at that lunch table, it hit him. Nobody had ever pointed that same method at a manuscript, at scripture itself. As it turned out, one particular fragment was made from cowhide. In fact, two fragments came back as cowhide. Together with Penina Shore, curator of the Dead Sea Scrolls at the Israel Antiquities Authority, Rechavi and Mizrahi assembled the small team that would spend the next several years chasing something nobody had ever attempted before. What they found along the way would shake even the people who'd devoted entire careers to protecting these fragile, irreplaceable texts. Reading genetics without a touch. Here was the first real obstacle, and it was a big one. The Dead Sea Scrolls are sacred to multiple faiths and, by their very nature, irreplaceable. Any method involving cutting, drilling, or removing physical samples was ruled out from the start. The scrolls had to stay whole and untouched. So, how do you read the genetic history of something you're strictly forbidden to touch? The answer had been there the entire time, hiding in plain sight inside the storage boxes themselves. Decades of careful handling had left behind fine particles of parchment dust, slowly collecting at the bottom of the containers. That dust held collagen, the structural protein in animal skin, and locked inside it, preserved across 2,000 years, was ancient genetic material. By collecting dust from blank margins and worn edges, spots where no ink had ever touched, the team could gather enough material without ever coming into contact with the written text itself. The evidence had been sitting quietly in that dust the whole time, through every study and every decade of scholarly argument. But turning that dust into usable genetic sequences was grueling, meticulous work. Making parchment, soaking, scraping, stretching, sizing, drying, damages DNA at a molecular level. The iron-based inks used on many of the scrolls are chemically corrosive, and on top of that, the scrolls had spent 2,000 years in desert caves before being handled by hundreds of researchers over the following 70 years, each one a potential source of modern contamination. The team worked inside dedicated ancient DNA facilities that looked more like clean rooms than archives, full protective suits, positive air pressure, UV sterilization. They built strict authentication protocols to make sure their results were genuinely ancient and not modern contamination, searching for the specific patterns of molecular decay that only accumulate over centuries, running samples through multiple independent extractions, and cross-checking every result across separate labs. It took years, hundreds of samples, and no shortage of failed attempts, but eventually they assembled reliable genetic sequences from dozens of fragments. And when Rechavi finally looked at what those sequences showed, he reached for his phone. The cow that broke the theory. Most fragments matched expectations without issue. Sheepskin, the standard material across ancient Judea, taken from an animal perfectly suited to surviving in a harsh desert environment like Qumran. But a handful of fragments came back as something else entirely, something that shouldn't have been possible. Cowhide Noam Mizrahi stared at that result for a long time. He knew Qumran inside and out, the texts, the terrain, the long, tangled history of the site, and he sat with the data in silence before saying anything to anyone. Because cow parchment at Qumran wasn't some minor anomaly you footnote and forget. It was physically, geographically impossible. Qumran sits in the Judean Desert right beside the Dead Sea, one of the harshest, driest places on the planet. Sheep and goats can handle that terrain. They're built for arid land, minimal water, and tough scrub vegetation that would starve almost any other livestock. Cattle simply cannot survive there. A single cow needs dozens of gallons of water a day, plus real grazing land, just to get by. There's no version of a community at Qumran that could have also been raising cattle nearby. Cow parchment could not have been produced in that desert. Full stop. Which meant any scroll written on cowhide had to have come from somewhere else entirely, somewhere with actual water and real pasture. It was wrong, and one cow made that undeniable. When that result landed on Penina Shore's desk at the Israel Antiquities Authority, she stopped reading and just sat with it. She'd spent her entire career as custodian of these fragments. She knew the dust in those storage boxes better than most people know their own homes. She read the finding twice, slowly, just to make sure she'd understood it correctly. She'd later describe it as feeling like the floor had shifted beneath her. This wasn't just a challenge to the Essene theory, it was a challenge to the entire framework scholars had built around what the Dead Sea Scrolls actually were. And the cowhide wasn't scattered randomly through the collection, which would have been odd enough on its own. It clustered around specific texts, specific versions of specific books, which is exactly where the genetics get even stranger. Fingerprints. Inside the parchment. Here's what the team could finally do that no one before them ever managed. By comparing genetic markers across different parchment samples, they could tell whether separate fragments came from the same animal. Two pieces of parchment from a single sheep carry nearly identical mitochondrial DNA. Two pieces from different sheep don't. For 70 years, matching fragments together had relied purely on guesswork. Now, for the first time, there was actual genetic evidence. Real fingerprints written directly into the material. Take the Book of Jeremiah as a clear example. Two dramatically different versions of it survive among the scrolls. One closely mirrors the familiar Hebrew Bible read today, longer, with chapters in the expected order. The other is noticeably shorter, missing entire sections, with chapters arranged completely differently. Before 1947, the earliest Hebrew manuscripts of the Old Testament available to scholars dated only to around 1000 CE. For decades afterward, experts argued endlessly over what these divergent versions meant. An early draft, maybe, or a later theological revision, or simply a scribal mistake. The DNA finally gave a concrete, physical answer to a debate that had dragged on for decades. The shorter version of Jeremiah was written on cowhide. The longer version was written on sheepskin. Different animals entirely, and almost certainly different regions. These weren't just variant copies that had gradually drifted apart from one shared original. They were two genuinely separate textual traditions, developed independently in two separate communities, in two different parts of ancient Judea. Read that again, because it matters. Two communities, two Bibles, existing side by side. The DNA showed that multiple Qumran fragments of this same text came from completely different animals. This text, it turned out, was popular, widely copied, and produced independently in multiple locations, not the exclusive guarded property of one isolated sect. Then came kinship analysis, which added yet another layer to the picture. By examining nuclear DNA markers, researchers could sometimes identify parchments that came from closely related animals, siblings, or members of the same flock. Clusters of related animals pointed towards specific production centers, places where several scrolls had clearly been made from locally sourced skins within the same narrow window of time. And here's the detail that matters most. Some of those tightly related clusters, obviously produced together from a single flock, turned up mixed in among fragments with no genetic connection at all, sourced from entirely different animals and entirely different flocks. This wasn't a collection that grew naturally out of one central scriptorium. It had been assembled from multiple points of origin, representing multiple communities scattered across the region, spanning multiple decades, quite possibly stretching across whole centuries. The library theory didn't just crack under that weight, it collapsed entirely. A world of many voices. Here's what all of this actually means. If the scrolls had genuinely come from one isolated sect living at Qumran, their sheer diversity wouldn't add up. Why would a single, disciplined community bother preserving competing versions of its own scripture? Why keep texts representing theological positions it actively rejected instead of simply discarding them? But if the collection had instead been gathered from many different communities scattered across ancient Judea, brought together in these caves gradually over decades, then finally sealed away during one catastrophic crisis, suddenly it all makes sense. The diversity isn't a complication that needs explaining away. It's the entire point of the collection. The genetic evidence backed up something a few scholars had long suspected but could never prove. These caves, the very same ones Muhammad ed-Deeb's stone broke open back in 1947, had functioned as a geniza, a sacred storage site where Jews from many different communities brought texts too holy to destroy, but no longer fit for daily use. Jewish law forbids discarding any document bearing the divine name. Such texts must be respectfully preserved instead. Ancient communities across the region kept designated spaces called genizot for exactly this purpose. Now picture the geography. Jerusalem sits barely 20 miles from Qumran, a manageable trip by ancient standards. When Rome laid siege to the city between 68 and 70 CE, Jews fleeing that destruction may well have carried whatever sacred scrolls they could save out into the desert, adding them to a collection other communities had already been depositing there for generations. Family scrolls, synagogue scrolls, fragments of an entire world being rescued as everything familiar collapsed around them. These caves, then, weren't the private archive of one monastic order living apart from society. They were a shared, sacred library belonging to the whole of ancient Israel. And the cowhide fragments prove something even more specific about how that library came together. Texts were physically traveling between distant regions, carried from wetter, more fertile land to the north or west into the heart of the Judean wilderness. This wasn't a closed, inward-looking community guarding its own private truth. It was a living, breathing network of Jewish communities exchanging texts, ideas, and religious practice across real physical distance. What this means for the origins of Christianity is genuinely significant. The world Jesus was born into wasn't one of settled, comfortable theological agreement. It was a landscape of vigorous, geographically scattered religious debate, with multiple communities each convinced they held the authentic tradition. The Dead Sea Scrolls weren't the position paper of one single faction inside that larger debate. They captured the entire argument in all its messy, contradictory detail. A Bible still being written for scholars of Scripture, the implications run even deeper. There was no single, universally accepted text of Jeremiah floating above the fray. There were multiple versions, shorter and longer and differently arranged, each treated as the authoritative form of Scripture by whichever community happened to be keeping it. The same pattern held for Genesis, for the Psalms, for the various prophetic books. Canonization wasn't a finished process handed down from on high. It was still actively underway, still contested, still genuinely unsettled. And now the DNA had proven that fact physically, not just theoretically. Separate communities in separate places were independently maintaining separate versions of what they each called the Bible. The textual tradition wasn't fixed or frozen. It was alive, evolving, and it looked meaningfully different depending on exactly where you stood within it. The world in which Jesus taught, and in which the earliest Christian movement first took shape, was steeped in exactly this kind of diversity. Competing scriptures, competing interpretations of the law, competing visions of what God would do next for his people. And the evidence for all of it had been sitting quietly in dust on a storage shelf the whole time, waiting for someone to finally ask the right question. Questions no one could ask before the 2020 publication of this genetic research opened an entirely new chapter in Dead Sea Scrolls scholarship, one that's still being written. What it made possible to ask for the very first time may end up mattering even more than what it actually settled. Before this work began, there was simply no way to scientifically confirm whether two fragments belonged to the same manuscript, whether a given text originated in one place or many, or whether separate scrolls shared any real physical connection beyond a loose similarity in subject matter. Now, for the first time, there is. Can researchers pinpoint specific production centers for individual texts? Can they map out the entire geographic web of Jewish scribal communities that once stretched across ancient Judea? Can they trace, in real physical detail, how textual traditions moved from place to place over decades or even centuries? Researchers are already chasing these questions, expanding DNA analysis to a much larger set of fragments, Pairing that genetic data with multispectral imaging capable of recovering writing invisible to the naked eye, and building reference databases of ancient animal DNA drawn from across the ancient Near East. The goal is a geographic map precise enough that a single fragment could reveal roughly where in the ancient world the animal behind its parchment once lived. The work of reading the Dead Sea Scrolls, in other words, isn't finished. In many ways, it's only just now truly beginning. Written in the skin, this genetic breakthrough is something considerably bigger than settling one narrow archaeological argument. For 70 years, brilliant, dedicated scholars studied these texts with every tool available to them, deciphering ancient scripts letter by letter, translating difficult and obscure dialects, painstakingly piecing together pages damaged by 2,000 years of decay. That work was real, valuable, and completely irreplaceable. But some questions stayed permanently out of reach, no matter how carefully anyone looked. Handwriting analysis is inherently subjective. Comparing parchment by eye alone is imprecise. Textual criticism can spot patterns in the writing, but it can't always prove for certain whether those patterns came from one community or from many scattered across a region. Molecular biology offered a genuinely different way in, one nobody had thought to try before that lunch conversation. By reading the genetic information locked inside the very material these sacred texts were written on, scientists finally answered questions that had once seemed permanently, hopelessly unanswerable. And what that evidence ultimately revealed turned out to be bigger than anything anyone involved had originally hoped to find. The Dead Sea Scrolls, it turns out, aren't the theological manifesto of one radical sect hiding from Rome in the desert. They're the accumulated sacred literature of ancient Israel itself, gathered piece by piece over generations, diverse, contested, deeply and messily human in its contradictions. Multiple voices, multiple communities, multiple versions of the Bible, each one carried into a cave in the Judean wilderness by someone watching their entire familiar world come to an end around them. We only know any of this today because scientists finally learned to read a story that wasn't written in the ink on the surface, but in the DNA of the animals whose skins carried that ink safely through 2,000 years of near-total silence. In 1947, a shepherd's stone shattered a clay jar and let an ancient world speak again for the first time in centuries. In 2020, a geneticist sequenced DNA pulled quietly from dust in a storage box. The shepherd opened the door. The scientist finally heard what had been waiting patiently behind it. Don't forget to like, share, and subscribe to Nature Mystery. Unravel mysteries, learn untold tales of history, and stay in the exciting world of new discoveries.